Welcome to Kim's Bio Production Academy. Today, let's understand of plant breeding. And this one I will major on the higher level. So this one will be students who are on the higher level careers. So from this one, first the thing that you should understand is about the plant breeder. Who is a plant breeder? This is a person. And who is this person? This is the person who concentrates on changing on a crop of variety, and this one is best so that it can adapt with the ecological conditions. So we are saying it's who? A breeder is the person. And this person concentrates on changing of a crop of variety, and the importance for the change of that crop variety is to adapt the ecological conditions required. I can use an example. In these crop varieties, we will have a crop which is based on resistance to disease. Others will be resisting to droughts, but in terms of the performance on the yield, it will be low. So what we should do is the plant breeder will be doing that. So they develop and come up with a new variety or cultivar. We combine various genes and we will get a very good. So let's say we have a crop with a good resistance to disease. We have another one which has a good resistance to droughts. We have another one which is good in yielding. But because of having something good, it will also have something which is negative. Let's say it's good in production, but not good in resistance. We take the gene for the resistance, we take the gene for the good yielding, we combine and we get a new variety. That's what we're calling, we get a new cultivar. From this point now, this one will be important because it will make them to respond to the inputs and even to the soils. Amelioration, which is necessity for food security and even income based. You understand our world's population is increasing. According to the statistics, it's being approximated by 2050 will be at 9 billion human population, and food security will be key in terms of the rising population. So we have to develop some cultivars which are high in production and even income based. Then they develop cultivars which are more efficient in water utilization. We're saying these cultivars, we develop our cultivars based on the following. First, water utilization. It should be efficient in water utilization. Should give us the first result when it gets water and also in limited water availability. Then number two, it should be a cultivar that these they can take maximum utilization of the fertilizer, either be it organic or the inorganic. When they're placed in the soils, or even if it's hydroponic, they should maximize the use of those fertilizers. Then the third will be able to resist the disease. So these cultivars that are being developed by the plant breeders, they should be tolerant to the diseases. Then they should be production or produce high dependable yields. The dependable yields should be very high, especially under minimum tillage. In what? In the dry agriculture or dry areas. You know, we have a lot of resting lands on the dry areas. Example in Kenya, the Turkana areas, the Marsabit areas. Those areas have vast lands, and these vast lands are just what? Inhabited by shrubs and those small plants and even the grasslands. We don't have good production in those sites. They are wasted lands. And so we are saying these cultivars are coming up with the developing of the dependable yield plants which can give us what? More production under minimum temperature, especially on the dry land. Then the last one, they will be able to resist pests and diseases. We are saying they can do what? They can resist the pests and disease and even the. So the significance for this one is just about the managing of the emerging issues with regards to climatic change, clean energy, and even food security. So we are speaking about what? Why we did varieties which were what? Well adapted to insect damage, tolerant to the diseases, and even water, and even maximum uptake of the fertilizers. So we are saying so that we can cap or manage the effect of the climate change. As you understand, in the world, we are experiencing climate change. And this climate change has been brought because of the green energy. Which one is this green energy? The accumulation of the carbon dioxide in the atmosphere has caused the greenhouse effect, which has led to the global warming. And that is why we are speaking about what? Climate change. And in relation for that, it will lead to the food insecurity. So we are coming up with this kind of a system in terms of the breeding so that we can cope or have plants which are well adapted to the climatic change. They can be well suited to the green energy, and plants which can have an adaptability of taking more of carbon dioxide from the atmosphere, and also which is high yielding to solve the problem of the food security. And this one will be important by innovating some skills to find solutions of the above mentioned problems. So the plant breeding will have some innovative skills that we're speaking about to solve of these what problems. So we have plant breeders. How will we be interested in developing of varieties which are drought? We're speaking about what? Tolerant to the drought. Plant breeders, how will they be interested in developing what? Which are the key features that we will have to check when it comes to developing a plant which is a drought resistant? And the first will be recognize the morphological traits. We are saying you recognize what was morphological traits. We have morphological traits and we have anatomy, the anatomical traits. So when you speak about the morphological traits, is what we feel and what we see. Anatomical is about the internal structures. This one is the external structures, of which the external structures you can check based on the leaf size, fruit size. That is what you can see. Then we have the physiological conditions, that is the functioning of that plant, and we have the pathological, that in terms of pest and disease response and even resistance in terms of that drought. So the response of this plant, they're important to adapt in terms of the yielding, quality of that crop, and even what? The adaptability of the drought conditions. So we are saying number one, when we are doing this plant breeding, the breeders are interested on checking, and we are starting number one with the drought trait. And on this drought trait, we are speaking about what? We check some morphological characteristics. That's the external. We check about the physiological features, and we check about the pathological. And this one will give us an adaptation to the yield and even to the quality. Then the second, evaluate now and identify the genetic potential. That is our second. So we are saying you evaluate and identify the genetic potential for the important trait in the crop of interest. As I was starting, I said our crop of interest here, we are speaking about drought resistant, right? But on the drought resistance, we'll have a plant which is well in terms of the resistance, but not good in the yielding. That's why now we are saying you evaluate. Evaluation, you do analysis of what and the comparison of various crop varieties. Then of that what? It's about the genetic potential, the genes that each and every plant will be carrying. Then from that point now, you search for the new source of genes. We are saying that second, you evaluate and you identify the genetic potential. Which potential does this plant have? Which is the potential that the other plant have? Then the third, you search for the new source of the genes. You search what? For the new source of genes. These genes, I've said in one plant, it will be having some gene for production. The other one will be having gene for disease resistance. The other one will be having the gene for high productivity. That is now where you search. So it means you have to go for another new set of a species for the desired trade of, for the drought what? Tolerance. And that may be utilized in the breeding program. So we are saying what here? You search for the new source for the gene. Our main intention is tolerance. So you look for drought tolerant plants. Then from that point, the last one, you combine now the genetic potential It's about combination of the genetic potential for these traits into the improved cultivar, the one now that you want to make. So we are saying you do some genetic combination. You combine one for the disease resistance, two for the drought resistance, three maybe even for what? Crop protection. And from that now you'll get an improved cultivar which will be well adapted to the drought conditions. Those are the plant breeder interest in developing of a cultivar which is a drought resistance. Again, now let's continue with factors that will determine the basic principles on population genetics. We have what? Factors that will determine the basic what? Principles on genetics. The first we will have mode of reproduction. Mode of reproduction. Second, we have random variation of alleles frequency. Third, we have selection for the mutation, and immigration being the fifth. Let's check about this mode of reproduction. On the mode of reproduction, you'll have sexual and we'll have asexual reproduction. When you speak about the sexual, this has to entail or involve about the gametes. And in plants, you'll speak about self-pollination and even cross-pollination. So that is what will bring about a new set of genes to a plant organism. So we are speaking about what? Mode of reproduction. It can bring about change when it comes to the plant breeding programs. Number two, we speak about variation. Parents, the parents will be sharing genes. So we have the parent, which is the male, and we have the female. And during the fusion time, we will have genes from one set and the other gene from the other set. And those genes will be in what? Pairs, which will be in allele form. And because of this one, they will bring about variation. And that is why the offsprings or the progeny will not be the same. They will just be different away from their parent one because of the combinations of the genes. Third, we have selection. Selection. A breeder will be selecting based on which characteristics will be suited to him based on the adaptation of interest. We're speaking about yield, the interest will be yield. So you do selection. And from that again, we have mutation. Mutation is just the spontaneous change. So we are speaking here what? Spontaneous change in genetic makeup. So this one is just a spontaneous change in genetic makeup. It occurs by chance. And because of this one, some genes we will have some mutation from gene mutation and even chromosomal mutation. So some genes might be deleted. Some might be substituted. Some might be inverted. So from those ones, we are speaking about the gene mutation. It can be gene mutation or chromosomal mutation. And if this occurs by chance, what will happen? There might be introduction of new genes. There might be elimination of other genes. And that brings about variation when it comes to the basic principles of the plant breeding methods. The immigration, this one is new set. New set of what? Of genes being introduced. Like in animal population, we speak about emigration and immigration. Emigration is moving out, immigration is coming. So we introduce another new set of genes, and this, according to the basic principles of population genetics, this will lead to the bringing about variation and even coming up with another new set of a plant organism or cultivar. We will not deal with genetics without naming the father of genetics, who is Mendel's Greco, an Australian monk. This person defined the population, and that population is a group of potentially interbreeding plants originating from one or more common ancestry. We are saying population is a group of what? Potentially interbreeding. plants originating from one or more common ancestry. So it's about potential interbreed of what? One or more plant species, but they will be originating from a same common ancestry. That is population. Then we have the phenotypic trait value. We have phenotypic and we have the genotypic. When you speak about phenotypic, is the perceived visual. When you speak about the perceived visual, that is what we see. In plants, we can use an example, the flower color. You can see the flower color either being red or being pink. When you check those two colors, you will be saying you are dealing with the phenotypic characteristics. Those yellow color, that white color, that blue color will be just an expression of the genotype. So the genotype will be internal. We'll understand that more. Then now we have the statistical analysis. What's the statistical analysis? These are used to test the hypothesis and the process data that help to adjust for variation. As we understand, we have types of variation: discrete, not discrete, but what? Null hypotheses. These now, we speak about what? They will be used to test for the hypothesis and process the data that help us to adjust about our variation so that we know to which extent does this variation brings about. So from that now, it will take us to our next step. We understand about the type of variations which can affect a trait. Types of what? Variation. And we just have two types of variation. And the first type of variation is the quantitative variation, and the second is qualitative variation. Quantitative and qualitative, sorry. This second one should be qualitative, not quantitative, but now qualitative. Qualitative variation. So the qualitative and the quantitative variation. When we speak about the quantitative variation, this one is variation that is about polygene control. It's about poly what? Gene. Poly means many. So it's the polygene control. It's controlled by many genes. But this other one, qualitative variation, is just under very control of major genes. In terms of a chromosome, you'll have genes. The genes will be inside a loci. So in that gene, we'll have many genes controlling a certain set of a genotype. And that is why now here we are speaking, when it comes to the qualitative, we just deal with the major genes. I can use an example of a height. You can either have plants being tall or dwarf. So that is major genes. Major gene for the tallness, major gene for the dwarfness. But now when it comes to the quantitative, we speak about many genes that control those factors. So, and that now takes us to those definitions of the gene, the loci. So let's get to understand about these definitions. We have the gene, we have the allele, and we have the locus, or you can speak about the gene locus. When you speak about the gene, genes will be located in the DNA. DNA will be located in the chromosome. So you start first with the chromosome. Inside the chromosome, you'll have a DNA. So we are speaking about this one. Let's say this one is our chromosome. This one is our centromere. This one is our sister chromatid. So inside at this point here, I'm saying at this point you have your DNA. That is the deoxyribonucleic acid. Then from that now point, we are saying inside this point, we have what? You'll have your gene. Inside now the gene, we are speaking about the alleles, the alternative form of those genes. Then now the locations. This one location where this will be found is the gene locus. So specific location along the chromosome. So if it's about at this end, that is the gene locus, that's the location where the genes will be located. Inside this point, I'm saying you have the gene, so inside the point is where we are speaking about what? So let's understand about the gene, the continuous region of DNA corresponding to one or more transcription. We have translation and transcription. How do we get this one? During the gene mapping, where we'll form a template with unzipping the DNA when we have the helicase or the enzyme polymerase, which leads to the unzipping of the DNA strand. Do you understand that DNA is in a double strand? So it unzips because of the presence of that enzyme helicase. And because of this one, we'll have two sets of what? We'll have some two strands, and this one will be made up of nucleotides A, T, C, and G. So we are speaking about the adenine, thymine, guanine, cytosine. These are nucleotides. So we are speaking about transcription units and consisting of a particular sequence of nucleotides. This is what I'm speaking about, nucleotide, and we are saying consisting of a particular set of a nucleotide. So this one is a continuous region of a DNA and corresponding to one or more transcription unit. The corresponding during the transcription, we get a template. This one is our template, and that is during transcription. And this one will be, so it's adenine. During transcription, we get thymine. Thymine, you get adenine. Cytosine, you get guanine. Guanine, you get cytosine. So this one is the one that has undergone transcription. That is what we are meaning about the genes. Then alleles, those are just alternative forms of genes. Alternative form. You understand about alternative? You can go this route or you can have another alternative way. Then locus is the specific location along the chromosome where the genes will be located. That specific location that these genes will be located. That is gene location. Let's do the last set here now. It's about, we speak about the phenotype. And based on the phenotype, this P is phenotype in quantitative variation. We discussed about the quantitative variation and we said those were which one? The quantitative what? Variation. The quantitative variation. We said they were polygene, which we mean they were controlled by various number of set of genes. And this one is a function. It's a function. The quantitative is a function of a gene and also it's continuous. That is the phenotype. Quantitative variation, function of a gene, and it's continuous. Phenotype, I said, it's just the visual acuity, the what we can see based on our eyes. So this phenotype is equals to what? Function of a gene. Function of a what? Gene. This P is equals to F gene. So its phenotype is equals to function of a gene. So in this case, genotype can be deduced from the genotype can be deduced from the phenotype. We speak can be deduced. This is what I mean. When you check a plant, like Mendel's used a pea plant. When you check that pea plant being tall, that is the phenotypic characteristics. From that you can deduce and know the inner plant section being having the genes which are tall. That is what we mean. P value is discrete. What's the meaning of it being discrete? It's distinct and what? Separate. That is what we mean. The value is what? Discrete. So it means it's just distinct. Distinction, we can separate tall, dwarf, blue, green, yellow, red. But that is what we mean. So it's the P value. That is the phenotype is always distinct. We can never confuse. It's either being tall or tall, dwarf or dwarf. That is it. Then, and the interaction of the two. So B, that is the phenotype, is equals the function of the gene, of which it will be both complex genotypes. It will be influenced by what? Both complex genotypes, those are genes, and two, quality of the growing conditions, that is the environment. So what will determine for us to get the phenotype is the genetic makeup of that organism or our plant, and number two, the environmental interactions. Then again, the interactions of the two. So it means it will be genes plus environment. The interactions, not plus but times. The interactions between genes, that is the genotype, and the environmental. So the last set now, the last thing that you just we have spoken about, you should know that this change absolutely important, and they are prone by the mode of reproduction, which can either be sexual or asexual, and the reproduction is absolutely important in determining the breeding of a crop. Crops will be cross-pollinated and they will be self-pollinated either sexually or asexually. Sexual way, we involve gamete. Asexual way, we have various. We can do budding, especially in the east. We can have fragmentation in case we break them. We have the budding, we have the fragmentation, binary fission, and even what? Sporulation. Thank you very much. That was our module 1 of the plant breeding. You can subscribe so that you can get more from the other sets. I'll be doing my second module, and our second module, if you'll subscribe, you'll get the notification and you'll get to learn more and more and more. Thank you very much for your time.